Hello, everyone. Hopefully, you can hear and see us pretty well. For everyone, we will still wait 5 more minutes, so everyone has the chance to join the meeting room. Plenty of you are already online, but we will still wait 5 minutes and start in for 5 minutes. Okay, I think a lot of people already joined, and the rest of them will be joining soon and will join the presentation of Max. So again, hello and welcome everybody to our third webinar of the Permeability series with the topic in vitro permeability experiments, experimental approaches and data interpretation and practice. My name is Yannick and I will be your host for this webinar. On today's agenda, we will take a closer look on topics that come up again and again when having meetings with customers. For the basic question on why to use in vitro tools to predict in vivo performances, to the explanation of an important indicator, the apparent permeability coefficient, and concluding with practical examples of experimental setups and corresponding interpretation of data gained. We are happy to again have Professor Massimiliano Pio Di Cagno, short Max, as our presenter for this live webinar. Max has a very impressive academic vita and can therefore be called a total expert in pharmaceutics and especially in drug formulation and permeability. Next to that, Max is also a freelancing scientific consultant for Fabiok in Germany, the company which manufactures and commercializes PermaPet. Max co-invented this product together with Professor Annette Bauer-Brandl back at the Southern Denmark University where he used to work. Before we start with the webinar, you have all been waiting for a few things regarding the organization. We have opened a Q&A window on the right side where you can type in your questions, which we will answer at the end of the presentation. And we will also provide the slides at the end and a link to the recorded webinar. Without further ado, I give the word to Max. Enjoy the presentation and see you at the Q&A session again. So Max, the stage is yours.
Thank you very much. I will then share my screen now. Okay. And I hope that you can see it. Everything is fine. So thanks to Yannick and to Fabio for having me again as a presenter. And the today topic is in vitro permeability experiments, experimental approaches, and data interpretation in practice. And this is the third webinar of the Permeability series we started early this year. The outline of the presentation of today, I will start with a general introduction of presenting why in vitro tools for predicting, for example, bioavailability and biopharmaceutics parameter could be very useful in drug development. And I will then show you some practical problems that arise generally when you perform permeability. I will focus on the issue on the unsteered water layer and related to the steering of the diffusion cells when you perform this type of experiments. And I will close with the selection, with the importance, highlighting the importance of the choice of the barrier. When you make permeability studies, you need to use a biomimetic barrier, but I will come back to that later. So, as a general introduction, I think it's very important to contextualize permeability testing and in vitro permeability testing into a pharmaceutical perspective. So, cost and time of drug development are rising exponentially in the last years. And when we consider the development, for example, a new API, so a new active ingredients, generally, a pharmaceutical company would start from a pool of 5,000 to 2,000 chemical entities to be tested. And from these, we will have just 200 to 300 compounds that will go from the drug discovery phase into the preclinical phase. And the one that will reach actually the clinical phase, so the latest stage of drug development, will be just 5. And generally, the statistics tell us that just 1 out of 20,000 compounds will become then an approved drug. This process takes circa 12 to 14 years, and it's very costly. Why it is very costly? It is very costly because the preclinical part of drug development, so basically where we are in these two blue phases from drug discovery to early early drug selection and preclinical research, um, takes approximately 1/3 of all the timeline of drug development, and it costs approximately the same as the clinical test. So it is extremely costly. And why this part, the drug discovery and the preclinical part, is so expensive? It is expensive because it relies basically on animal experiments. In vitro testing is used industrially, but still limited, and generally it is not used to select the best candidate as it should be yet, but it is mostly used for in the process of filing for approved drug, because this is demanded by the FDA and EMA. But we need to change the things. This should be exactly the opposite. In vitro testing should be implemented to screen the drug and to limit it to the most to limit the most possible animal testing. So in vitro testing, how do we see the future for in vitro testing for biopharmaceutics and bioavailability? Definitely, they will reduce the cost and time of preclinical drug development. They will also allow the step forward in mechanistic comprehension of permeation and absorption that still is lacking. And very important, they can provide useful parameters that still are lacking for in silico drug profiling and modeling. And of course, all of this should be contextualized in the goal of the implementation of the 3Rs principle: refine, replace, and reduce animal testing, that is actually into the European legislation since 2010. So soon enough, will not be allowed anymore to utilize in EU and USA approximately 100 million animals per year. This will be not allowed anymore. So we have to find appropriate solution and SOPs. One of the solution could be to implement more the measurement of the so-called upper permeability. The upper permeability is a parameter that is experimentally measured, and is measured with the permeability assays. All these assays relies basically on diffusion chambers, small, like in there could be 96 well plates, they could be larger, like the front cell diffusion cells, but the system and the physical description is the same. We have a donor compartment where the drug solution or the drug formulation dispersion is placed. Then we have a barrier, a biomimetic barrier, and then we have an acceptor compartment. Between the donor and an acceptor exists a gradient of concentration that we should try to keep as constant as possible. And mostly the receptor is often steered, as well as the donor. If we use a system that can be placed on an orbital shaker, for example, then we can steer both compartments. If the concentration gradients remain constant during the experiment and we properly perform the experiment, we can measure the flux, so the accumulated mass over time in the separate medium. Normalizing the flux over the concentration of the drug in the donor, we detect the so-called upper permeability coefficient. The unit of the p-up is a centimeter of a second, is a velocity, and you can directly compare, if giving for granted that you have utilized the same type of barrier and assay, you can compare this parameter with drugs and permeability testing that has been performed in different labs around the world. This is true for permeability. And why this permeability is a crucial parameter? Because already in 1995, when Amidon et al. Introduced the BCS classification system, it was highlighted the importance of the measurement of a permeability through, in that case, it was considered the intestinal epithelium. But contextualizing nowadays, we could say that if you are capable of measuring an upper permeability coefficient through a biomimeric barrier that is capable to mimic the behavior of the intestinal walls, the gastrointestinal walls, then measuring the permeability will immediately indicate the theoretical bioavailability of the compounds after administration. And the challenge or the possibility that PermePed allows is not only to measure the API and predict its bioavailability, so its fraction absorbed after oral administration, but it also would allow the testing of enabling formulation. That is a crucial challenge in drug development nowadays. Concerning the experimental setup where the PermePed barrier can be adapted or other barrier, generally we have, as I said, a donor compartment, a barrier, and an acceptor media. We can have macroscopic diffusion cells, like the front cell diffusion cells, but some of this setup, like the PAMPA, the parallel artificial permeation assay, and the PermePed can be are available also in 96 well plates for increasing the high throughput screening ability. Other type of diffusion cells are also side by side, where, for example, the Hussin chambers, where actually the barrier is not placed horizontally, but is placed vertically. But the concept is the same. And here you can see a figure for the one that of you that have not experienced working with it yet. This is a figure of the PermePed in its PETS format, the first development product that, from Fabio. This is the barrier as it looks like, and this is the barrier that is placed, for example, in a front cell to measure the experiment, to run the experiment of permeability. This is a fully functional biomimetic barrier that doesn't need to be activated. It is ready to use. And nowadays, already the barrier is also available in the 96 well plates format, where each of the wells will represent a small diffusion chamber, so a small experiment. Of course, the plate is ideal when you need to test in high throughput screen, so you need a lot of results in the shortest time possible. In terms of practical in the experiment of this kind of experiment, what we do is basically measure the accumulation of the drug in the acceptor media over time. So if this figure represents the acceptor media over the time, you can see that the blue color darken itself. That means that the drug slowly accumulates into the acceptor media over time. And what we practically do in the experiment, we measure a flux. So we calculate the amount of drug Q over the surface area of the barrier that has accumulated over time. And when should we actually interpolate this curve? This experiment generally are subjected to a so-called lag time, so where the barrier needs to get saturated and reach a constant concentration of the drug. This can be very long in some setups, but for PermePed this is very, very short, or even absent for highly permeable compounds. Then we enter in the so-called steady state region of the flux, where the permeability, where the accumulated amount grows linearly over the time. And this is the part where actually we are allowed to interpolate and with a linear regression get the slope of the curve. The slope of this curve is the flux that we actually measure. Put it representing the same figure with another type of diagram, we will see that the flux get constant after the lag time, and this is what we are interested in to find out to measure the permeability. The flux cannot be directly compared because the flux is proportional to the gradient of concentration, so the concentration of the drug in the donor. So if we divided the flux over the concentration of the drug in the donor, we get the permeability. The permeability is, and I repeat it, is a velocity, centimeter of a second. And this, assuming that we have tested the permeability of these compounds with the same setup, with the same type of barrier, can be compared with the results obtained in different labs all over the world. Of course, to get the permeability, we always have to keep in mind, and this is true for any permeability studies, that we are making some assumptions. The first assumption is that we consider the diffusion as a diffusion in an homogeneous media, and this is not really true because we go from a aqueous environment through a lipoidal environment, through an aqueous environment again. Then we have to be able to hold the concentration gradient constant. In this way, we can actually rewrite the Fick's law as a diffusion coefficient multiplied by the concentration gradient, so the difference between the concentration of the drug in the donor and in the acceptor, divided by the thickness of the barrier. And then we also assume that the concentration in the donor should be much higher of the, comparison to the concentration of the acceptor. This allows to simplify even more the equation. And I think you can see that here we have our permeability, because if we move the concentration from this side, then we come to the same equation. So P up can also be written as the diffusivity of the drug divided by the thickness of the barrier. One practical aspect that can be interesting is that is interesting to underline is the maintenance of the sink condition. One question I often get is how can we assure the sink condition? Well, there is not a real thermodynamic laws for this, but there are some rule of thumb that are generally accepted. The first one is that the concentration of the drug should not exceed the 5% of the solubility of the drug in the acceptor media. And the other one, and this is the one I actually follow the most, is that the accumulated material should not exceed the 5% of the total amount of the drug in the donor. Why that? Because if this is happening, we have too much drug going through, and then instead of having a nice and long line, we have a bending of the curve. We have a reduction. So we have a very short steady state where we actually don't really know if it's correct, and then we have a curvature. I'm saying that if you're very good in mathematics, you can definitely utilize also this kind of curve, but this is not simple, because then you need to fit a non-linear regression curve with several unknown. First of all, the gradient of concentration. So my suggestion is always try to hold the concentration gradient and obtaining a nice linear regression. This simplifies a lot the measurement of the permeability. In practical, how do we maintain the sink condition? If we have macroscopic diffusion cells, what we do is we sample from the acceptor port, and then we replace the volume of the sample with fresh buffer, for example. When we utilize multiple plates, we can do the same. What we actually do in my lab as a routine is also to do the same. So we remove some volume from the acceptor and we replace it with the new material, but we don't have a robot. If you have a robot and you have availability of different plates, then you can just move your upper donor plates to a new acceptor media every 30 minutes, for example, and reading this with a UV plate reader, for example. Another interesting aspect is to understand when we might have some problem with experiments. For example, the most utilized approach when we make permeation study with the front cell of other vertical diffusion cells is to place the formulation on top and sample from the bottom. But for the one of you that has worked with the front cell of other diffusion cells, you might have setups that are actually quite long. So you can have like online 6, 7, 8, 10 front cells. Oh, and what this will bring? This will bring that you can have a concentration, a temperature gradient between the first cells and the last cells of approximately 1 to 1.5 degrees. And this will affect the permeation of the drug. So it's very important to remember this, to account for it. So my suggestion would be, for example, not to use all the cells, but to use maybe 3 or 4 of them, and to have a thermometer in one of the empty cells to monitor the temperature gradient and how this will change over the time during the experiments. Because the temperature is a parameter that can affect the permeability. Another important thing is, another problem is the low detectability of the drug. And this is especially true when we have macroscopic front cells, like the microscopic diffusion cells, like the front cell. So one alternative that can be used is and it's very suited for PermePad because PermePad is a non-polarized barrier, so you can use it in both directions, is to place your donor solution in the bottom and sample from the top. In this way, you have an acceptor volume that is very small, and this will allow the concentration of the drug in the acceptor, making the analysis much easier. Of course, that also might not solve the problems from time to time for very poorly permeable compounds. So what should you do if it does not solve the problem? Then you can try to make the acceptor media maybe a little bit more, pass me the term, lipophilic. So if the drug is very lipophilic, then you can add, for example, some organic solvent, 5 to 10% of methanol, ethanol. We tested also cyclodextrins are very good to increase the flux, the albumin could be also a possibility. But in some case, this might not also be sufficient. So another and last alternative I would suggest is to change the analytics. UV is, of course, the first one you should try, but from time to time, it would not be sensitive enough. So you can go to HPLC and move forward to fluorescence, to LCMS, according to the analytics you have in your laboratories. And here is an example of actually unpublished data we obtained with my first PhD students back in 2013 that helps in the PermePad development. And we tested hydrocortisone, that is a lipophilic drug with a log PA of approximately 2. Low soluble compounds, 0.9 millimolar, so not terrible. And when we tested this with a front cell, 8 milliliter front cell, in the beginning with a up to bottom approach, we could not detect any measurable absorbance. So we decided to use the bottom up approach, and actually we actually need also to dilute the sample to get it, and we could actually easily measure it. Calculate the concentration of the variation of the accumulated mass over surface area in the acceptor media that basically can come from the interpolation of the flux of this mass transfer profiles, the one I showed before. So the slope of this curve we represented the flux, and then dividing the flux by the initial concentration of hydrocortisone, that in this case was 0.9 micromole cubic centimeter. That is, it was the measured thermodynamic solubility of hydrocortisone, because for very poorly soluble compounds, it's a good idea to work at thermodynamic solubility to achieve the highest gradient possible in terms of concentration. And then we measure the permeability. So you see that the permeability were actually pretty close. Just to remember that this barrier we utilized were actually handcrafted. It was still in the development phase. Now the barrier has a standard deviation that is much lower than 5%. Another important aspect to account for, and it's definitely not taken into consideration often, is the composition and the properties of the donor solution, because we always focus on the property of the API we want to investigate. But when you test enabling formulation, or when you actually want to mimic even closely the GI absorption, now we have available a lot of we have actually the possibility to utilize, for example, this passive and passive and biomimetic mimicking substances. And of course, these substances will alter some important properties of a donor, for example, the viscosity. So that's why it's important to consider the experiment and to consider that unstable water layers, so stagnant layers of water that would lay on top and on the bottom of the barrier might be present during your experiment, even though you stir. But this is not an issue. The important thing is that you remember this and you account for it, or try to account for it, especially when you have a high viscous solution in your donor compartment. Because if we try to describe this with in terms of concentration gradient, in the donor solution we will have a certain volume where the concentration will be constant, but then close to the barrier we will have for sure a small thin layer of water where actually the diffusion of the drug play the crucial role, and we will have a concentration gradient. And the same, even though smaller because of the steering, in the receptor media. And how much the composition of this donor could affect was and investigating how much the properties of the donor solution could affect the permeation was the focus of our latest work here at WIO. We tested again hydrocortisone, and we decided to test hydrocortisone in PBS solutions, so phosphate buffer saline, in a concentration below the solubility and at the thermodynamic solubility. And then we utilize 3 different PEG with different length, 400, 3000, and 6000. But we choose the concentration to hold the viscosity at the same level. So the two solution in aqueous media has a viscosity of circa 1 millipascal per second. The pegylated solution has a viscosity of circa 6 times higher. Note that with all the PEG, we obtain a supersaturation. So we highly increase the molecularly dissolved drug concentration for hydrocortisone. So expecting a higher permeability. So if we actually look at the experiment with the PBS, the pink one represented the undersaturated solution of hydrocortisone, and the blue one, the saturated solution. We get a small variation, not a very significant one, but this is expected because when you measure the permeability, you account for the concentration. So it is quite normal that these values are extremely similar, even though there might be some variability in terms of microviscosity and whatsoever. But the very interesting results are the one related to the viscous solutions. So if we have a solution with a viscosity of 6 millipascal, it is the same in the 3 different donor solution, we have a huge reduction in the net permeability. So this is extremely interesting because the permeability is the interaction should be the reflection of the interaction of the drug with the barrier. And what these results tell us that viscosity has a huge impact on this measurement. But this is also happening in real life. A recent publication from Müller et al. Shows the importance of considering the viscosity when preparing enabling formulation or enabling formulation. And the second thing that I would like you to notice is that this experiment allowed you anyway to find out some features of each of the polymer. So if we consider these polymers not as viscosing agents, but as a possible enabling formulation, we would see that, for example, the PEG 6000, the supersaturation effect is significant, whereas for the PEG 400, this is not significant. And this is exactly what we are interested in when we are testing enabling formulation, to look for these differences, and especially for observing or having an assay that will tell us if a formulation is able to achieve a better drug permeation and expected oral bioavailability compared to the available formulation. So this is definitely something that PermePed can give it to you. But always, when you make permeability studies, you always should take care of the viscosity, because this allows you to normalize your results. And also this allows you to understand the mechanistic, what is happening. And what we clearly saw is that with a small PEG, there was a close interaction between the polymer and the drug, and this was making macro complex with a lower diffusion, in comparison to when we have larger PEG, where the diffusion is more a percolative phenomena, and then with hydrocortisone being a lipophilic drug, the interaction between hydrocortisone and the polymer is not that significant. And then another possibilities is, instead of using the classical diffusion cells, is to use what I would call flow-through diffusion cells. These are extremely useful because especially when we have issues with the austere water layer. So if we see that the austere water layer can be a problem, and we are testing enabling formulation, and we are not so much interested in the high throughput screening, then we can utilize this kind of cells. We tested the MIVO from React for Life, and they actually work brilliantly. In this case, you again have a donor compartment and an acceptor compartment where you in the middle place the barrier, but in the receiver chamber you don't have a stagnant liquid, but you have a flow through, so that you have a constant flux of fluids that of course remove the unsterile water layer. And this is a huge advantage because you can measure the flux, but this basically remove both the lag times and also remove the effect of the unsterile water layer in the acceptor media. And what we find out, for example, testing the MIVO with hydrocortisone, again, we use a lot of hydrocortisone because it's an extremely good model drug. If we measure the permeability of hydrocortisone in 2 different pH using the plate, we get not very significant, but systemic difference between the permeability at pH 2 and 7.4, where pH 2 hydrocortisone resulted lower permeable. But then when we actually tested with the MIVO, this permeation, this difference was completely lost, telling us that in hydrocortisone permeation, the unsterile water layer and the acceptor media, even though we steer, can play a minimal but systemic role. And the last things I want to point out today in this presentation is the choice of the barrier. In some reports, the literature is written, for example, that size exclusion barrier like regenerated cellulose can be used for mimicking the permeability or for studying permeability of drugs in vivo. I don't really agree on that. If you want to measure the permeability and to have a proper prediction of the permeation, you need to utilize a barrier that has some biomimetic properties. A regenerated cellulose or dialysis barrier in general should be classified as size exclusion barrier because, as you can see from these 2 different setup of experiments, where several drugs have been tested on permeability utilizing Franz diffusion cells and multi-well size diffusion cells or another comparable system, you see that the permeability is proportional to the molecular weight of the drug. So to say that larger is the molecular weight of the drug, lower is the permeation measured with this dialysis barrier or regenerated cellulose. And this is not the case. This is just passive diffusion. But because in this type of barrier, there is nothing biomimetic, there is no lipid, there is nothing that mimics the interaction of the drug with actually the cell monolayers when we talk about permeation. Another important limitation of using this barrier is that you are not allowed to make an experiment in pH gradient. And this is fundamental. When you want to test the absorption of the drug through the GI, you have to test these at different pH. In the stomach at pH 1, and in the stomach intestine pH 8 from 7 to 8. Whereas the acceptor media, so the part of the diffusions that mimic the blood, should be always a neutral pH. So if for proving that, we utilize again the MIVO, and we utilize regenerated cellulose as a barrier, and we test ketoprofen. Ketoprofen is an acidic drug, and as all these non-steroidal painkiller drugs of acidic nature are generally quite nicely absorbed already at acidic pH, because at acidic pH they are neutralized, and the neutral form of the drug has a higher permeability than the anionic form. Testing with the MIVO, what we get is actually that the permeability was higher at pH 7.4. Testing the MIVO and the regenerated cellulose, what we find out is that we got a higher permeability at pH 7.4, which is not biologically true. And this is just because at pH 7.4, the drug has a much higher solubility than the acidic form, and therefore you have a so high concentration gradient that it affects the net permeability. Testing, on the other hand, the same drug in the same condition with the PermePed, we got actually a real physiological interpretation. So permeability of the drug that was 20% lower in neutral pH than acidic pH, and this is exactly as it should be. We tested also a basic drug like atenolol, where we saw this the same behave, but in the opposite direction, where the permeability was much higher in a neutral pH, because this is a basic drug. So that is very important. When you select a barrier for permeation testing, you need to select a biomimetic barrier. And this is it. This is what I wanted to tell you today. And thank you for the attention. And I will be here for answering to your questions.
Thank you very much, Max, for the presentation and all the insights you gave to us. We already have 2 questions from Gautam, and I received 2 questions in the private chat. But Gautam, the first question: Hello, Max. Could you please shed some light on the cell line which was used in PermaPet? Maybe I can answer this, or I understood this question that you want to know which cells are in PermaPet. But actually, this is a completely artificial barrier. It is just cellulose and a lipid layer in between. There are no living parts or cells in our barrier. So it's completely artificial, and it's just a phospholipid.
But if I can add something, what is happening upon hydration is that in the B layer, liposomes are forming. So basically, you have a reassemblement of a cell layer without, of course, the nuclei and all the components. So that's why it's so extremely biomimetic.
And that is just useful for the passive absorption. So there's no active transport at all with PermaPet. Okay, the next question from you. What, according to you, Max, can be the mechanistic explanation for the high difference observed between the undersaturated versus saturated condition while using PEG 6000?
This is a good question. This comes from the different concentration. I can. Is it the presentation still visible?
Yes,
Yannick?
it is.
Okay, so I just show you the diagram again. So if you look at the table on top, with the PEG 6000, we got a higher concentration for the supersaturated solution. So we have a circa 40% higher concentration than in PBS. That is why we have this difference. For getting this data, we normalized over the total amount of drug. So it's difficult for us to know what was molecularly dissolved and what was actually a reservoir of the drug, but what we see is that this different, what we believe is that the difference comes from the highest concentration gradient generated by the enabling formulation.
Okay. If you have further questions, please feel free to
Absolutely.
ask again, Gautam. The next question is from Philip. How did you come to use maximum of 5% of thermodynamic solubility for zinc conditions?
Maybe I want to rephrase. So this is for the septomedia. So to hold the same condition, my suggestion would be, especially for a drug that might be poorly permeable, is to try to be careful with how much drug permeate, because you can also have recrystallization, etc. So a parameter could be 5% of the thermodynamic solubility, not to overcome to this. This is what is generally reported. This is a parameter that I not always hold. I'm focusing more on the septomedia and not to have a higher concentration or a too high concentration compared to the septomedia, more than on the thermodynamic solubility.
Okay. Again, here, if you have further questions, please feel free to ask. Then the question from Mudin. Hopefully,
Mhm.
this is right spelled. How Permapet is useful in generic product development, and can it be used to predict permeability using USP dissolution apparatus, basket, or pedal?
I think maybe Yannick, you should answer to this question, but it's possible. Yes.
Yes, it is possible. There are several devices on the market, for example, from Electrolab or Logan Instruments, or the Macroflux from Pion, where you can integrate the biomimetic barrier. So to simulate the dissolution and permeation process in one assay. So yeah, there are several apparatus on the market, and they show quite good results. It's not too easy because you have large volumina, but of course we are happy to help or assist you in experimental setups and data interpretation. This is what we offer with Max together. Yeah, to have a successful essay in your research and development. A further question from Mudin. Is there any variability or special concern with drugs belongs to different BCS class and with respect to the um, MFG technology, for example, solid dispersions or hot melted?
I don't know if I get completely clear the answer, but I would say that the standard deviation is generally very low, also when you test enabling formulation. Of course, the experiment need to set up according to your needs and what you want to investigate, but there is the standard deviation is generally quite acceptable. Yes.
Is this useful for you, Mudin?
Yeah.
Okay, great. Now to the questions from my private chat. How can I measure the drug resistance through the unstirred water layer?
Well, there are different ways. The easiest is to measure the diffusion coefficient. And if you know the steering rate, you can have an estimation of the thickness. And with these two parameters, you can get the resistivity or the permeability of your drug. So the easiest would be to have the diffusion coefficient of the drug, and then you can account for it. Because the larger is the lowest is the diffusivity of the drug, the highest is the resistivity through the unsterile water layer.
Okay, I think this might be useful. Um, and then would the 96 well plate be as resistant as the barrier in macroscopic assays, such as diffusion chambers or front cells, whatever?
I would definitely so, from my experience, absolutely.
Okay, pretty easy answer. Uh,
We got also another answer question on the chat.
yes,
Yes.
yes. Yes, we will send you the link for the recording. It will take, I think, 3 days, and then you will receive the link from the Webinar Ninja, and then you can download and the recording or have again a look on the video. Furthermore, I just unlocked the slides, the PDF you can download right now. Those are the slides Max just presented to us. Oh, I just changed the presentation again, the layout. So I haven't received any new questions yet. Are there open questions? If you have afterwards any questions, please feel free to contact us. I just type in an email address where you can get access to us, .com, and then we will find a solution for your problem or issue, and we will be happy to assist you in any ways. So if no question arise in 5 seconds, I will close the webinar and stop the broadcast. Max, thank you again for your
My
great
pleasure.
presentation,
Thank
for
you
your
very much for having
insights,
me again. It
and
was a pleasure.
For the Permeability series, we will have a summer break, and we are working hard on getting external partners to present here in this series. For example, the paracellular transport or the use of PermaPad in the different mucosa. But we will inform you shortly about everything through our LinkedIn account. So please follow us there, and you get updated pretty soon with new webinars of the Permeability series. Thank you very much for your time. Have a great day and see you soon. Bye bye.
Likewise. Bye bye.